Today I will be speaking to you on comprehensive antibody characterization using intact mass and bottom-up enhanced top-down mass spectrometry. Pretty much I will present on this combined approach that our lab uses to enhance our antibody characterization. I will take you through a series of different antibody characterization of varying complexity. So for a brief outline of my talk, I am going to introduce the pharmaceutical life cycle, where our services, our CRO services fit in. Then I will give you an overview of our workflow using intact mass, top-down and bottom-up mass spectrometry. Then I will present some data using this multi-level approach, focusing on 3 case studies, starting with our simplest case, a monoclonal antibody, the USB MAP3 reference standard, a monoclonal antibody mixture, and bispecific antibodies. And then I will summarize and leave the floor open for questions. Let me get my pointer. Okay. So here are the 5 stages or general stages of the biopharmaceutical life cycle. So you normally start with your target identification, which is where you would pinpoint some specific biological molecule implicated in a disease that you would target for treatment. You would optimize that molecule, and then there's quite a bit of characterization done at this stage. Following this, there is preclinical research that involves in vitro and in vivo testing in animals. This is for safety assessment and toxicity studies. Following this, you'll have your clinical trials. That generally includes 3 phases, 1, 2, and 3, where the drug is tested for safety, its dosage and efficacy, and there is continuous patient monitoring here. And for all these stages, your data is submitted to regulatory authorities for review. And if successful, you will get a market authorization where you're able to market your drug, and it can be used in general public. And then finally, there is commercial production, quality control, where and real-world data monitoring, as well as safety monitoring. Our lab falls within the first 3 stages, where our primary focus is the discovery and development stage and any R&D stage, and non-GMP as well. And so, since the talk is about antibody, how does this life cycle look for antibody? You would start, like as previously mentioned, with your target discovery, ID, and validation. These are the initial phases where, again, you pinpoint your biological molecule responsible for a disease that you could target with your antibody or antibodies. This is followed by the lead discovery and selection phase, where you generate a large pool of potential antibodies against your validated target, and systematically narrow down a few leads that could with the best therapeutic potential. When you found that molecule or molecules, you may need to modify its structure for maximum therapeutic effect. And all of this falls under the discovery and optimization phase. Then, once you have your optimized molecule, you would seek to create stable, high-producing cell lines as host cells that can manufacture antibodies. And finally, you would need to scale up from a small laboratory approach to a large-scale approach, and this falls under the development and manufacturing phase, according to general CMC guidelines. And our lab supports characterization at any one of these stages. Of course, there are overlap and primarily non-GMP work. And so some of the critical quality attributes that we're focused on for antibodies includes looking at structural integrity. We're experts at sequencing. We also do product purity and heterogeneity, looking at different variants, whether your antibody is mispaired. I will show an example of that at the end of this talk. We also look at post-translational modifications, glycosylation profiles, and degradation products. And this is not an exhaustive list. So to give you an idea of the workflows that I'm going to present, they are LCMS and LCMS-MS workflows, and it includes bottom-up, top-down, and intact mass analysis. And so let me walk you through each one. So our bottom-up analysis, I should mention that all these experiments were done on these high-resolution instruments, Orbitrap Fusion Lumos and Orbitrap Exploris. With various fragmentation techniques, including ETD, ET8CD, 8CD. And so to start with, the bottom-up approach, it's your standard approach. We would digest, and for our approach, we use a multi-enzyme digest to enhance our sequence coverage. Usually there's some form of sample cleanup. We do our MS-MS analyses. We have many software tools. Some of them are released as software, some of in-house that allow us to do our peptide sequencing. And this would be for both database searches as well as de novo sequencing that would allow us to assemble our intact molecule. At the intact level, we do this as 3 levels, whole, reduced, and subunits. So the whole could be, we do both with glycan and then deglycosylate. Any data are presented in deglycosylation. The glycans were removed with PNGASF. For the reduced level to generate a light and heavy chain, some reducing agent like DTT or TSEP. And at the subunit level, for all the data presented, we use IDZ to cleave the hinge region, and then we reduce to get the light chain and heavy chains. And so the fragments that we end up with would be the LC, the FD prime, and the FC/2. And then we would analyze this directly on our instruments, and then followed by deconvolution to get our all intact species. At the top-down level here, which is sort of a bridge between the bottom-up and the intact to get proteoform level information and modifications of proteoforms. The experiments presented in this work were done at the subunit level. And the fragmentation technique was ET8CD. And so all of the data obtained from these experimental workflows was analyzed with our software recently released, Proteoform X, except for instances where I did use specific in-house software which I will mention as the data is being presented. So the first case study I want to present on is a monoclonal antibody, or simplest case in this presentation. It's the USB3 mAb. It's a highly characterized antibody reference standard with a certificate of analysis purity reporting 99.6%. This is sort of a proof of concept that our method works. So we chose something that is highly characterized. You've got your 4 polypeptide chains, 2 heavy chains here, your light chain. It has 16 disulfide bonds, single N glycosylation site in the FC region at asparagine 297, and then you have N-terminal pyroglutamate and C-terminal lysine deletions as modifications, as will be determined in the next few slides. So for the intact work, in terms of our software, this is Proteoform X. When you open Proteoform X, you will see several workflows here. Since we're doing intact mass, that's the one that's selected. You would pull in your raw data here, select your instrument. In this case, it's Orbitrap, and the molecule at what level you're analyzing. In this case, it's whole, but there are other options to look at reduced species and subunit species. Then you would set your deconvolution parameter to get your zero charge mass spectrum and the mass of all species that's present. In this case, I've got, for the whole, it's set at 50 part per million error, mass range of 120 to 160 kilodaltons. There you can set a retention time range if you've got chromatography. And then in this case, I've used a sliding window of 0.1 minutes. I've set it to annotate the species detected for everything above 5%. And I'm searching against, it's the USB3 mAb. I've pulled in the sequence here. You can do this. You can select your sequence in two ways. You can paste it in, or you could pull it in from Proteoform Manager in the software, and you would have to define it in the software. That's what it looks like. You pull in the sequence. You've got the light and heavy chains. You can set your disulfide bonds and any modifications. The colored bits here are modifications. The asparagine 297 is here. That's the glycan. And so for some results, for intact mass analysis at these two spectra at the top here, it's for the whole molecule. At the bottom, it's for the whole and the deglycosylated species. On the left is our raw data. On our right, our deconvoluted spectra. If you look at the raw data here, you see multiple species. At the whole level, the deglycosylated species, there seem to be disappearance. In order to know what they are, of course, you deconvolute. So if we look at our fully intact molecule, we see there are 5 major glycoforms detected. So we're able to resolve those species. We're also seeing lysine clipped here. This is the species that we detect are comparable to what is in the literature for this molecule. If you look at the intact deconvoluted spectrum of the deglycosylated forms, no glycans are detected on the species as expected. It's predominantly your deglycosylated intact molecule as expected. At the reduced level, at the top here, I've got the deconvoluted spectrum for the reduced species. The bottom, it's the deglycosylated species again. You have your light chain here. Here you have your heavy chain, and we see the heavy chain is glycosylated as expected, and we're able to localize lysine clipping on the heavy chain. For the deglycosylated versions, the light chain is detected. There are no glycans, of course, on the light chain, so it's same as the intact here. The heavy chain is deglycosylated since there are no glycans detected. And so once again, at the reduced level, species detected is comparable to what is published. At the subunit level now, so we've treated our molecule with IDZ to cleave the hinge region, and then we reduce to obtain our 3 subunits here. These two spectra at the top are with glycans, at the bottom, deglycosylated. You have your light chains for both with glycan and without glycan. And then you see the other 2 subunits as well. We've got the FD prime and the FC over 2. The FC over 2, as expected, is glycosylated. And then here it's deglycosylated species as well. Sorry, it's deglycosylated sample, and we don't see any deglycosylated species. So we're able to confirm that we're able to detect all species for the USP3 map that's comparable to the literature, and we're able to localize chain-specific modification by looking at it at the reduced and subunit level. So now if we wanted to characterize this at the proteoform level with top-down mass spectrometry, this is how we would do it. In this case, it's top-down of the subunits, so technically middle-down mass spectrometry. So once again, we open our Proteoform X, select our workflow. In this case, it's top-down. We pull in our raw data, select our instrument, select the type of fragmentation. We use ETHCD here, whether we expect our isotopes to be resolved for our species of interest. And then you select all your top-down parameters. You're able to put the charge, mass error, the signal to noise that you look at for the MS1 here, the MS2, the mass error tolerance, in this case is set to 10 ppm. Then you pull in your modifications. We have some default modifications that are grayed out here. For this specific molecule, we know that there is lysine clipping. I've included the usual oxidation. And then there is pyroglutamate formation from E that is expected. You can also set the maximum PTM expected per proteoform, I've got that to 5, and then there is this open PTM search that is open. You select the mass range within which you want to search. Then I've set the top-down score here for it to report everything that's greater than 1. So the results starts with a summary page here. You have notes, you have some stats, you have all your parameters that you have selected. One thing I want to point out here is that you see we've got 22 proteoforms, but 3 proteins. So these are the 3 subunits that underwent the top-down experiment. And so if we look at some data, this is what the top-down results window looks like. I don't have everything shown here, just the ones that I'm using. So you have your proteoform, the entire sequence of what is detected, the observed mass, the intact level, the expected mass, your mass error, any modifications. These are sequence coverage at the top-down level, top-down scores to indicate how confident you are of the data, and the species of interest that was detected, got retention time, mass to charge of the species, the charge, and the protein spectrum match ID. Below here, it's the protein spectrum match. So this is the fragmentation spectrum that was linked to the intact proteoform. So for example, if we take this proteoform here, it's the FC/2 subunit. And these are the fragment ions used to confirm its presence. And you see we've got coverage right across the entire molecule here, with a sequence coverage of 87.08%. Now, our software has had several improvements, and we continually improve our software. But one of the things that allow this high sequence coverage, it's the software combines multiple scans to enhance the coverage. And so if we look at all of the data, so this is data for non-targeted top-down of the 3 map subunits. You've got your light chain, your FD prime, and your FC/2 region. On the right here, it's the corresponding spectra that was used to match to the protein. First, I want to point out that we've got coverage across a lot of the intact proteoform. And we're able to detect the light chain at 76.9% coverage, the FD prime at 87%, the FC/2 at 87.1%. We can also do these experiments for a targeted approach. Here we selected the top 6 chart state above 20 for top-down fragmentation. On the left, we've got a coverage maps. On the right, we've got the fragmentation spectrum of the intact proteoform that we use to map to its proteoform. And so we've got the light chain, the FD prime, the FC/2 subunits. These were all detected with high confidence, and these measurements for the targeted approach were comparable to the non-targeted approach. So we're able to garner proteoform information that is linked to our intact information. Now, if we wanted to, the title of this talk is bottom up enhance. So how do we enhance our top down with bottom up measurements? You it's the same workflow that you use, your top down workflow. You pull in your top-down data as before, but notice the bottom-up tab. And here you would pull in all your bottom-up data that you want to search together with your top-down. And I previously mentioned that we use a multi-enzyme approach. So here we've got multiple enzymes, as well as we've done both ET8CD and 8CD fragmentation to enhance our sequence coverage. You pull in your top-down parameters. These are the same as used before. You pull in your bottom-up parameters here. I'm working with a precursor mass error of 10 ppm, fragment ion error of 0.02 Daltons. You select your PTMs. So there's a fixed modification of carbamidamethylation. And then we've got all of the variable modifications expected, and I've set this to 2 PTMs per peptide. And the top-down score for reporting is the same as before. Now to look at the combined data. So this is data for the USB3 mAb at the subunit level, top-down and bottom-up combined. So you've got the LC chain, the FD prime, and the FC/2 subunit. At the top here, you've got the top-down coverage. And at the bottom here, it's the overlapping coverage for the multi-enzyme approach. So each one of these bars is a peptide. Each color is a different enzyme. And for this particular molecule, we're able to get 100% sequence coverage. Now, for this study, remember, we're characterizing our model system, and it does have a glycosylation site, of which the most abundant glycoform, it's G0F and G1F, on the intact protein. And so we decided to use a combination of top down or middle down, because we're targeting the subunit, and bottom up to characterize our glycan. So for the top-down measurements, here at the top, you see the top-down coverage. We've got the 70% coverage. The bottom up, we've got 100% coverage. This is targeted, again, for the FC/2 subunit that we were able to measure at the proteoform level within 6 parts per million. If we zoom in here, we see that we do have the glycan localized on the FC/2 subunit at intact proteoform level, and we have it also on various peptides from the bottom-up data. If we take one example, here's the glycopeptide fragmentation spectrum that we use to confirm the N-glycosylation site. And so just to give you a bit more detail, so that's the spectrum here. You've got your diagnostic oxonium ion indicating that this is indeed a glycopeptide, one of many diagnostic ions present. This is the glycan fragmentation spectrum. We see sequential fragmentation of the glycan. This is the glycopeptide backbone fragmentation, and we see great good coverage across the peptide molecule. The B ions that were detected were all non-glycosylated, so we used the Y ions from C-terminal fragments to delineate the site. So, for example, we've got a Y9 here that is glycosylated. We also have the Y2 that's not glycosylated, but the Y4 that's glycosylated, confirming that site. And for comparison, if you look at the non-glycosylated peptide backbone, we see that Y9 without any glycans. So we're able to characterize our model system, indicating that our method works. So for my next case study, this comprises an antibody mixture with the USB1, the USB2, and the NIST mAb. These are all highly characterized species and sort of serve as our model for co-formulated mAb cocktails. For example, a cocktail of casirivimab and imdevimab was used to neutralize SARS-CoV-2 during the pandemic. With more complex systems, there is need for analytical platforms as well as software tools, of course, capable of resolving complex overlapping proteoform landscapes. And for our studies, and our services, we have many critical quality attributes that we can measure, and as presented before, this includes your amino acid sequence verifications and just sequencing an unknown species, molecular mass, impurities, glycosylation profiles, PTMs. Now to look at some data. So this is data for the mAbs in a 1:1:1 ratio. At the top here, it's the intact spectra for raw data on the left and deconvoluted spectra, the deconvoluted spectrum on the right. At the bottom, like the USB3 mAb, we've got the deglycosylated versions. Of course, if you look at the raw data, it's complex. You've got overlapping signal. So when you deconvolute, if we look at the whole intact species, we see many species. These are all glycosylated. They are resolved for the 3 antibodies, so we're able to resolve the glycoforms. And here you've got the USB1 mAb in green, the NIS mAb in blue, and the USB2 mAb in orange here. So we're able to detect glycoforms at the intact level. If we look at the deglycosylated spectrum, once again, we're able to see all 3 antibodies resolved without any glycans. For the reduced sample, once again, at the top here, we've got the glycosylated verse sample. At the bottom, we've got the deglycosylated sample. We're able to see all 3 light chains, and we're all able to detect the other 2, the heavy chain. The heavy chains for all 3 species with lysine clipping, localized, paraglutamate formation from G, as well as glycoform. So at this step, once again, we're able to localize that glycan on the heavy chains of these 3 antibodies. Looking at the deglycosylated versions here, you see the 3 light chains for the 3 antibodies, and you see the 3 heavy chains. So everything is in line with what is expected. If we look at the reduced subunits, the subunits that were treated, the antibody treated with IDZ and reduced, we've got our 3 subunits. You've got the glycosylated sample at the top, the glycosylated samples at the bottom. We see our 3 light chains. We see several species for all 3 maps, and the chains are the FC/2 and the FD prime chains. And note that the NIST and the USB2 are not resolved for the FC/2, and that is because they have the same sequence. If we look at the deglycosylated version here, we've got the 3 light chains for the 3 antibodies, and we have 5 instead of 6 subunits here, once again, because the NIST and the USB2 match share the same sequence. This exemplifies our multi-level approach because we're not able to resolve them at this level, but we are able to separate them at the heavy chain, at the reduced level where we see the 3 heavy chains, and at the intact level. And if we want to look at top-down characterization, to look at this at the proteoform level, we've got this is the top-down sequence coverage maps obtained for the antibody mixture. Firstly, we're able to detect all 3 antibodies in the mixture, varying proteoforms for these antibodies. And so at the top here, you've got the USB1 mAb, the light chain, the FD prime chain, and the FC/2, the USB2 mAb similarly, and the NIS mAb here. And so we're able to characterize this at the proteoform level. And so what if we were to go deeper and combine this with the bottom-up data? So here once again, we've got this is the USB1 mAb in the mixture. At the top here, you've got your top-down coverage. Here, it's the bottom-up data or multi-enzyme approach. As you can see, we have 100% sequence coverage here. So highlighting the approach, the multi-level approach. Similarly, for the USB2 mAb, we also have 100% coverage as well as the NIS mAb. But really what we wanted to know in looking at mixture analysis is that if the antibodies are not present in equal ratio, how sensitive is the method for detecting lower abundant components? And so we did our top-down analysis as well as our bottom-up analysis for 3 different antibody mixtures. So we have our USB1, USB2, NIS mAb at 331, 551, 1010-1. Here are all the subunit chains detected. We are able to detect all subunits regardless of the ratio used here. That's the sequence. It covers the entire sequence. We've got our observed mass and the expected mass all within 40 part per million. We're able to characterize this at the top-down level, getting coverage ranging from 14% to 67.9%. And so we're able to, for these experiments, we're able to detect all our subunits regardless of ratio with bottom-up coverage of 95 to 100%. So the lowest we're able to go for these experiments was here, which is the lowest component was about 5% of the other components. And so this brings me to my last example. It's our bispecific antibody. So a bispecific antibody is an engineered structure that can bind to two different antigens or epitopes simultaneously, unlike traditional monoclonal antibodies that bind only to one. One of the strengths of using high resolution mass spectrometry is that you are able to solve the mispairing problems. You can distinguish the correct heterodimer from homodimers and impurities, of course, by measuring mass at part per million mass accuracy with a multi-level approach, as previously shown. So, for example, this is a correctly paired bispecific. You've got 4 different chains, 2 different light chains, 2 different heavy chains. Mispair products would include homodimer formation and light chain scrambling. So homodimers meaning that you've got the same heavy chains and the same light chains. Light chain scrambling, you've got mismatch of your light chains. Looking at these molecules, given that they're asymmetric, looking at them at the subunit level with both intact and our middle down approach, you were able to localize specific modifications, otherwise indistinguishable at the intact antibody level. So you can say what modifications are there on specific arms. And of course, you can, if you need to verify the sequence or validate the sequence, there is the bottom-up approach that confirms that we can confirm our antibody sequence up to 100%. We can also localize amino-specific modifications with looking at the peptides directly, as I will show you. And of course, our in-house algorithms using de novo sequencing are invaluable tools for identifying novel sequences and unknown variants. So for my first example, of a bispecific. It's mosunetuzumab here. It's an asymmetric bispecific antibody with 2 distinct arms, one for binding CD20 and one for binding CD3. Note that the FC region has a mutation that has removed the asparagine, so that's usually glycosylated. So there are no glycans present, or no N-glycans, I should say. And so if we were to look at the intact mass of mosunetuzumab, so the sequence is available. We've got a chain A, heavy chain, a chain B, a light chain, a chain C, another heavy chain, and a chain D, a light chain. So 4 different light chains. At the intact level, the most abundant species here is it corresponds to the expected molecular weight for the mosunetuzumab with 2 lysines clipped, 1 lysine clipped, and the intact mosunetuzumab. So we're measuring the expected mass for the most abundant species, but we also have another species present here, which I will come to later on. If we were to look at this at the reduced level and the subunit level, so at the reduced level where we expect to see the 2 light chains and the 2 heavy chains, we are seeing that. So you've got your chain D and chain B, which are the light chains, and you got your chain A and your chain C which are the heavy chains. All confirmed within 27.4 part per million of the expected mass. At the subunit level, we expect 6 subunits. So that is what we got, 6 main subunits. You've got your chain B and D, which are the light chain. You've got the FC/2 and the FC/2 for both C and A. So this is the heavy chain split in 2. This is you've got your 2 light chain, and then you've got your FC/2 for each heavy chain, and the FD prime for each heavy chain. So we've got our 6 expected subunit. But really the question we want to answer is it paired correctly? And so this is our PolySeq AI in-house software that I use to do this analysis. So you would input your intact, reduce, and subunit data into here, and then you would do your pairing analyses. And so what you see here, it's two heavy chains, two light chains. This heavy chain, I neglected to mention, this species corresponds to this species that you see here by molecular weight. But so you've got your heavy chain. The first one, you've got chain C here. You've got another heavy chain, chain A. You've got chain D. You've got chain D. All measured within 37 part per million here. And from the pairing analysis, this chain C comes from the FD prime and FC2 subunits here. This other heavy chain comes from these two subunits by mass. And then there you have your two light chain. And if you were to sum the heavy chains and the light chains detected here, that equates to a correctly paired species. So the most abundant species is the correctly paired species, what we expected. If we go to the other main species detected, it would be this species here. From the pairing analysis, we've got chain C, or heavy chain, chain A, chain D, which is a light chain, and that's the only light chain that was detected. Of course, the chain C was put together from these two subunits, the chain D from the chain A from these two subunits, and then you've got light chains here. If we were to sum this, we would end up with mispaired species here. Actually, it's actually light chain scrambling. So you've got, instead of this species here, you've got the two heavy chains paired with the light chain. So we were able to confirm the correct pairing solely by mass for our most abundant species here, but there was also another species, about 25%, that was at about 25% relative abundance that was detected. And so are we able to characterize these at the proteoform level with our top-down data? Yes, we are. Here are the 6 subunits. This is done at the subunit level. We're able to use our top-down data to map to these intact proteoforms, confirming them within 11 part per million mass accuracy. Our top-down coverage range from 34.6 to 84.7. And the heat map here shows you the distribution of the 6 proteoforms, their elution profile, retention time versus mass. In the interest of time, I will move through this quickly. So these are the combined top-down and bottom-up data, with the top-down spectra showed at the bottom here, where we have coverage across a lot of the intact molecules here that we were used to get our top-down data that would allow us to do our characterization at the proteoform level. And so we were able to confirm everything's present with the combining bottom-up and top-down data. And so now for my last example for a bispecific, it's a bit more complex than the previous one. It's catumaximab, which is a trifunctional and bispecific antibody. Trifunctional is that you've got three binding areas, you've got the 2 arms, and this Fc region also is functional. Interestingly, it was removed from the market in 2017, but recently relaunched in February 2025 with market authorization in the EU. There is no sequence available for the product that we worked with, so we started this with bottom-up. Our lab was able to construct the sequence for all the chains. So here you've got the light chain. This is the bottom-up data, or multi-enzyme approach with the overlapping coverage. You've got the mouse light chain, the rat light chain. You've got the mouse heavy chain and the rat heavy chain. These are what we expected. If you From the sequence that we constructed, we're able to get the molecular weights, and so we're able to do our intact analysis to compare if we've got the species paired correctly. And this is the proteoform manager sequence inputted with modifications and linkages, and this is for a mouse-rat hybrid. And so if we look at our intact mass analysis data, the most abundant species here, we've got proteoforms and glycoforms, but instead of our mouse-rat hybrid, it's predominantly mouse-mouse. So it's not the most abundant species is not what we expected. What we expected is actually present in lower abundance here, where we do see a mouse-rat hybrid, and we've characterized this at both the reduced level and at the subunit level to confirm what is there. But we also saw extensive glycosylation, and we wanted to characterize that glycan. Can we localize it on a specific chain, and can we localize it on a specific site? So we did bottom up as well as middle down, targeting various FC/2 subunits where we expect glycosylation. I'm only showing you one example. So for the top-down data, the proteoform that we targeted was the FC/2 subunit, measured at the proteoform level within 5 parts per million. The glycan expected was this A1G0F glycan. Our top-down coverage here was 72.3%. Our bottom-up coverage was 100%. Since this is about the glycan, if we zoom in here, we do see a glycan at the proteoform level from our top-down data. We also see glycans on peptides from different digests. And so, as an example, here is the glycopeptide that is glycosylated. If we look at it in more detail, you see the glycan sequenced here. If we look at the glycopeptide backbone, we see coverage across most of the backbone. All the B ions, your N-terminal sequence ions, are non-glycosylated. So for diagnostics, to determine whether the site is actually glycosylated, we use the Y ions. And so the Y2 from this end and the Y3 are non-glycosylated. But if we look at the Y5 and Y4, they do have glycans, confirming that this site is glycosylated. So that brings me to the end of my talk. In summary, integrated approach was presented that utilized intact mass, bottom-up, and top-down mass spectrometry for comprehensive antibody characterization at different levels of structure. Our software, Proteoform X, and others in-house are powerful tools for this combined analysis, particularly using bottom-up and top-down together to link proteoform level information with peptide level information. The analyses provided detailed sequence coverage up to 100%, PTM identification, and localization at different levels of structure. High mass accuracy measurements and microheterogeneity assessment for a monoclonal antibody reference standard, an antibody mixture, and bispecific antibodies was also presented. We identified bispecific mispairing with the molecular differences between the desired heterodimer and unwanted homodimer by looking at intact mass data at different levels of structure. And overall, our methodology can be applied to other systems such as antibody drug conjugates or antibody oligonucleotide conjugates, which we're currently working on. With that, thank you for your attention, and I will take any questions.